I'm Paul Mischel. I'm an Institute scholar here at ChEM-H and a colleague of our next speaker, Lingyin Li. A personal story, when I first came to Stanford 2 years ago, I was talking to our beloved and sadly departed colleague, Chris Walsh, and he said to me, "You need to meet Lingyin Li. What she's doing is really important," and Chris was right. I think we're going to hear something really, really interesting. By way of background, Lingyin did her PhD at University of Wisconsin, where she really began to work on chemical biology. And then she went to Harvard Medical School, Tim Mitchison's lab, really at the interface of chemistry and biology. And I think that served her very well because her work right now is shedding new light actually on the immune system, really taking a chemistry-first approach. We're learning something quite fundamental, actually, from Lingyin's work that really promises to transform, I think, our understanding of some of the processes of evading the innate immune system that goes on in cancer and how to circumvent this. She's won a number of awards already early on in her career, including a Talented 20 from Chemical and Engineering News, an NIH New Innovator Award, and the Eli Lilly Award in Biological Chemistry. It really is a pleasure to introduce our next speaker, Lingyin Li.
Thank you, Paul, for bringing us back to the Chris Walsh era. And without him, I wouldn't have been here. And it was his vision and Chayden's vision that I was hired. I remember when I was hired, my postdoc advisor, Tim Mitchison, was so proud and surprised. He said, kudos to ChEM-H. A person named Lingyin Li is getting a job. So it's a curse to be a chemist because we can work on any biological problems. So for me, I didn't have a set vision when I started at my postdoctoral position. I wanted to study the brain. I wanted to know how learning occurs and how neurons fire. And I was interested in neurotransmitters, thinking a chemist could do something about neurotransmitters. And then I was diagnosed with breast cancer myself when I just turned 30. So that sharply focused my own research and now my lab's research. So if you find my focus incredibly narrow, I'm sorry. So I'll just say that this was 12 years ago now. And as you know, breast cancer patients, what you get is debulking, okay? So surgery. For anyone, right, who is experiencing this, I would recommend surgery. No doubt, it's not medicine, right? And then after that is whether it's cytotoxic drugs or targeted therapy, debulking is definitely the number one. I'm not going to say, let's try immunotherapy. But after you've tried all that and you still want to do better, okay, that's when we want to try immunotherapy because immunotherapy holds the hope to clear the very last cell, right? Just like how we clear viruses. So of course, I asked my oncologist, my doctor, whether I could benefit from the drugs that just came on the market at that time, Keytruda as well as Yervoy. And she said, no, they don't work in our breast oncology ward, and we don't know why. So now, of course, it's known knowledge that common knowledge that most solid tumors, breast cancer, pancreatic cancer, glioblastoma, prostate cancer, they simply don't have these green blue cells infiltrated, so you can unleash their power all you want. With Keytruda, they're not going to work. So then our question was, how do you infiltrate these killer cells? Okay, so the understanding is that cancer cells are fundamentally different. So how can we, just let them attract these blue-green cells? Is there an innate immune signal, which is the most upstream of immune detection of any abnormality? So long story short, this is not our research at all, right? This is the collective effort of the whole field of cancer biology, immunology, biochemistry. We now know that a cancer cell is fundamentally different in that they cannot keep their double-stranded DNA in the right place. Okay, and the mechanism is very long, and including chromosomal instability, or the mechanism that is elucidated by our own Paul Mischel and Howard Chan, that X-chromosomal replication of oncogenes. So when double-stranded DNA is detected in the cytosol, it is then turned into a second messenger called cyclic GMP-AMP simply by cyclizing ATP and GTP. And cGAMP then binds and activates a protein called STING, which stands for Stimulator of Interferon Genes, which by its name activates interferon production, which is a very potent attractant by itself to the brain blue cells. Okay, so when cGAMP was discovered at that time, 12 years ago now, immediately people think, let's use cGAMP to activate human immunity against the cancer. But there's a problem because cGAMP is a second messenger, and all second messengers are degraded very quickly. So I purified, doing what chemists know how to do, using activity-guided purification, purified its dominant hydrolase, and turns out to be a known protein called EMPP1, and E stands for ecto or extracellular. So that created a problem because cGAMP is intracellular and its dominant hydrolase shouldn't be extracellular. So easy explanation is I was wrong. That's very possible. Alternatively, it is because cGAMP is exported out. Because I was thinking about a neurotransmitter, so it was not difficult for me to hypothesize that cGAMP is pumped out by cancer cells. And Stanford ChEM-H is one of the very few places that gave me opportunity to test its hypothesis. And Jackie Carrozza, our first-year CBI student, who was sent to me by Chetan Khosla, saying, you got to give this young professor a try. So she used a mass spec to definitively show that cancer cells indeed pump out cGAMP. They not only always make cGAMP, they then pump it out at a rate that every 2 days they clear all the cGAMP they make. Okay, and great, so cGAMP is detected. So what? We're showing that, sorry for summarizing her work in just one little figure here. She showed that ionizing radiation, which can reliably cure a substantial amount of tumor-bearing mice, is doing so by generating extracellular cGAMP. Because when you remove the extracellular cGAMP, which jumps from the blue line to the red line, then all the curative effect we can observe in mice disappears. Okay, so this tells us that now perhaps ionizing radiation can be combined with immunotherapy because it is activating cGAMP production and is activating the downstream STING interferon production. So now, if natural cGAMP pumped out by cancer cells is already detected, is sufficient to be detected by the host, which host cells? Again, using a chemistry approach, we simply ask, we know the host cells, when they're detecting cGAMP, it has to phosphorylate its STING, has to phosphorylate its downstream transcription factor and interferon. Then if we remove cGAMP, which cells no longer do so? This is one example of a cell type, which is the M1 macrophage. You can see when you remove cGAMP, in the red dots, you see a decreased STING phosphorylation, decreased phospho-IRF3, decreased interferon production. So with this type of chemical approach coupled by just flow cytometry, we were able to find out these are the direct responsive cells to extracellular cGAMP, macrophages, tumor vasculature, T cells. And then if you do a time course in a few days, they then infiltrate the blue-green cells I was showing in the very first slide and activate them. Okay. Overall, cGAMP produced by ionizing radiation is able to give you that immunologically hot tumor that should respond to the current immunotherapy. So this is great. Then why doesn't ionizing radiation work in people just as well as it does in mice? It occasionally does have curative effect, and when it happens, it's called Oboscopo effect, named after Dr. Oboscopo, but it's so rare, okay? So maybe I'll skip this. The reason is because that 50% of tumors overexpress ENPP1, and the more metastatic they are, the higher ENPP1 they would express. Okay, so now ENPP1 is perhaps the reason some cold tumors are cold. Here's some human data, not our data, publicly available. Whether you can mine it on Biomatic, you can mine it in TCGA. You can see that patients, this is breast cancer patients, patients with high ENPP1 expression live on average 5 years shorter. Their disease-free survival is much shorter. Okay, and what that means for breast cancer patients when they have primary surgery, right? That means metastasis. Okay, so then we looked at ENPP1's role in metastasis, and you can see that these are the breast cancer staging. In stage 4, ENPP1 level is the highest. So this is human data. It's difficult to do experiments in humans. Can we do it in mice and mirror the results, the phenomenon we're observing in people? And the answer is yes. So Sonan in the lab, she overexpressed either live ENPP1 or enzymatically dead ENPP1 in the blue curve. And you can see that primary tumor growth, there's a little bit of difference. It's not earth-shattering. What is earth-shattering is metastasis. Only breast cancer cells overexpressing enzymatically live ENPP1 metastasize into the lungs, which is a common place for, in mouse models at least, for breast cancer metastasis. Okay, so then this is when, you know, when you're at an institute where just being a chemist and just being a cell biologist, just being an innate immunologist is not going to move the needle for human health. You actually need to now do single-cell-seq. So we performed single-cell-seq analysis of the primary tumors as well as the metastasis to see what is the cellular or signaling explanation of this metastasis promoted by live ENPP1 expression. So I don't mean that you should look at the dots, but here, just to summarize that tumors expressing live ENPP1 primary tumor-wise as well as metastasis have less T cells infiltrated, have less B cells infiltrated, and have less T cell activation marker. So that's just bulk cell count. And then if you look at what signaling they're doing, you can see that when the tumor is overexpressing ENPP1, that's a wild type. That's the pink bars you're looking at, violin plot you're looking at here. They overexpress arginine 1, for example, and arginase 1. Arginase chews up arginine, and arginine is important for T cell metabolism and killing. Okay, so this is how they dampen T cell function. And ENPP1 wild type expressing breast tumors also have less dendritic cells and less antigen presentation. And as a result, they have less T cell activation shown in the first 2 T cell activation markers and have more T cell exhaustion shown in the next 2 T cell exhaustion markers. Okay, so that seems to be in line with their ability to degrade cGAMP and therefore dampening interferon. So far, we're focusing on cancer ENPP1. In our single cell analysis data, we also noticed that not just cancer cells express ENPP1. The stromal cells and infiltrating immune cells also express ENPP1 and in an inducible fashion. So, for example, fibroblasts which is known to perform a wound healing kind of role for cancer and promote tumor growth, has a lot of ENPP1, as well as macrophages. And we noticed a stepwise increase of ENPP1 in macrophages from the most immunostimulating M1 macrophage to the most immunosuppressive M2 macrophage. You see their ENPP1 increase and their interferon signaling decrease in the same way. So that seems to tell us that they are directly internalizing cGAMP, and how much ENPP1 they have is their gatekeeper of how much cGAMP they internalize and how much STING interferon they turn on. So this is exciting to us because that seems our original hypothesis that host cells, immune cells take up cGAMP seems to be shown here. How does this happen in all the host cells? So remember, this is single cell data. So we could look at every single cell and look at their ENPP1 expression level, and then look at how much interferon they make. And you can see that this is when ENPP1 is highly expressed in a cell, their interferon stimulated gene, IFIT-Ms, are the lowest expressed gene. So this seems to be, is exciting to us for every single cell. How much ENPP1 they have dictates how much cGAMP and how much STING they activate. Okay, so I'll just say that the slide I skipped over was a part list over the years that my lab put together is, for example, all the transporters of cGAMP, and every single one of them was painstakingly tested, and of course using chemistry and biochemistry tools, and I'm sad that I have to skip that. But then now with this single cell data set, we can look at each transporter and can look at their roles, and everything seems to be checking out. So it was quite an emotional day for me to see our transporters being mapped onto the recipient cell types. So, but this tells us that the host ENPP1 plays just as big a role as tumor ENPP1 themselves. So then it's true. If you now count ENPP1 in the cancer, the tumors, primary tumors decrease a little bit. Now count out in the host, decrease a little bit. Now counting both, tumor is the smallest, but still not earth-shattering. What is earth-shattering to me is metastasis. What you're seeing here is at the end of the experiment, we take out the lungs, and then we would kill all the normal cells. The cancer cells are drug resistant, as always. So what you're seeing here is the drug-resistant cancer cells. When you inject wild-type cancers in wild-type animals, but if you now count ENPP1 in the mice, you see less lung metastasis. You now count ENPP1 in cancers, you see less metastasis. You now count in both, you do not see metastasis. So now this is great, right? But there's a problem. ENPP1 now count animals do have a phenotype, and that is artery calcification and joint calcification. They get skinny over time. So when they have a smaller tumor, when they don't have lung metastasis, is it because they have super immunity, or is it because they're simply too skinny to grow tumors in their lungs? So you do have to rule out that possibility. ENPP1 has two different substrates mainly, okay? Even though I discovered its role in degrading cGAMP, therefore dampening STING signaling, ENPP1 was known to degrade extracellular ATP, okay? So we need to differentiate these two functions. So Jackie and her undergrad, Yasmin, identified a mutation that renders ENPP1 not able to degrade cGAMP, but still has full function otherwise. And Anthony made a mouse. This mouse does not have joint calcification, suggesting it's not its cGAMP hydrolysis activity that causes this phenotype. Okay? So now this mouse runs freely, gets just as healthy and as fat as a primary, as a wild type, so we can study its ability to fight metastasis. wild type, you see metastasis. STING knockout, you see metastasis. So some people start to say, oh, STING doesn't play a role in metastasis. It's because there's ENPP1 that degrades all the cGAMP. In this mutant mouse, that ENPP1 cannot degrade cGAMP, you do not see metastasis. When you knock out STING, metastasis comes back, okay? So this tells us that it's really the cGAMP hydrolysis activity of ENPP1 that is promoting metastasis. So I will skip over. I cannot skip over this. I'm sorry. The single most important slide. If everything else I said is not necessary. So chronologically, this was the first thing that happened to us, okay? So when I started at ChEM-H, I was sent, Daniel Fernandez was sent my way, and Mark Smith was sent my way. I thought I was attracting them. No, I'm pretty sure the leadership is sending them my way and say, you gotta help this helpless person. So with their effort that we developed ENPP1 inhibitors, okay, and you know, this inhibitor actually helped our analysis of extracellular cGAMP along the way. This is our secret sauce. Without it, we would not be able to detect cGAMP. And this inhibitor is going to clinical trials. But maybe I'll just last say that now, with Hao-Ching Wang's help, we were able to develop, in collaboration with Jim Wells, a cGAMP-specific neutralizing antibody of ENPP1. So showing here that it only inhibits cGAMP hydrolysis, does not inhibit ATP hydrolysis. And indeed, it is behaving just as well as the total ENPP1 neutralizing agent. So this antibody itself is not going to be a therapy. It has many problems, but it just shows that it is possible to develop such an agent. Moving forward, the vision is that when a cancer patient's metastasis can be treated, then the patient would be on this therapy for decades, hopefully. So we really need something that's very clean, only targets cGAMP hydrolysis, doesn't give breast cancer patients calcification on their joints, right? So that we can all live happily ever after. So thank you all. Sorry about overtime. Yeah. I might not have time.
Do we have time? Sure. Okay, one question. It was a wonderful talk. Question?
Great.
You got one shot.
This is such an epic story. And one question that comes to mind is that ENPP1 is membrane associated, right? So it's on the surface of cells. And do you know anything about its turnover dynamics? And also, is it shed? And do you find it free in circulation ever?
Yes, it is. It is not shed on the cell membrane. It is 50% of ENPP1 is actively secreted into the plasma, which does control the systemic metastasis. So that also needs to be controlled. Mm-hmm. Yeah. WE.
What about concentration in lymph nodes? Do you know about that?
Concentration of ENPP1 in lymph nodes. It's high because it's the highest ENPP1 expressor is actually B cells. Yeah. Okay, thank you.